Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, please, if you can, take a moment to like, subscribe, and share this with your friends and family. That small action really supports the Science Experience and, helps us keep bringing you these complex topics. Today, we are going to be diving deep into one of medicine's biggest challenges: repairing the heart after a heart attack or a myocardial infarction. When an MI happens, the body tries to fix it, but, instead of growing new muscle, it just creates this stiff, useless scar. And that scar tissue, it doesn't contract. Over time, that's what leads directly to chronic heart failure.
It really is the ultimate failure of regeneration in mammals. I mean, in most mammals, including us, we just can't replace lost heart muscle. The ambition of this research, and really the mission of our deep dive today, is to explore something called direct cardiac reprogramming. It's this revolutionary idea of taking those scar-forming cells, the cardiac fibroblasts, and forcing them to become functional heart muscle cells. They call them induced cardiomyocytes, or iCMs.
Now, we sort of know the basic recipe for this kind of magic trick, right? It relies on a trio of transcription factors. It's the classic MGT cocktail: MEF2C, GATA4, and TDX5.
That's the one.
And these factors, you deliver them with a virus, and they're supposed to just flip the cell's identity switch.
That's the key. Supposed to. The MGT cocktail works, but here is the massive clinical hurdle this paper really gets into. See, DCR works great in neonatal mice. Their cells are still really plastic, but the efficiency it plummets when you try to apply it to adult or, and this is critical, aged hearts. So our goal today is to unpack the specific molecular blockade that aging puts on regeneration. And reveal a single, high-impact target that could unlock the heart's own ability to repair itself.
Okay, let's start with the hard numbers then, the ones that really paint a picture of this age barrier. When the scientists looked at cells from newborn mice versus older mice, the difference was pretty shocking. What did that baseline data tell us?
The difference was it was massive. In the neonatal cardiac fibroblasts, using that MGT cocktail gave them a successful conversion rate of about 15%.
15%. So 1 in every 6 cells basically became a new heart muscle cell.
Exactly, which is an incredible starting point for this kind of work.
But then they moved to adult fibroblasts. So cells that are comparable to a human in their 20s or 30s, and that efficiency, it just fell off a cliff, didn't it?
Oh, completely. It dropped sharply to less than 3%. And when they used aged fibroblasts, from mice equivalent to elderly humans, the efficiency was, it was barely scraping 2%.
Wow.
So yeah, that basically told them that just tweaking the recipe wasn't going to cut it. The cell itself was inherently resistant because of age.
So the question becomes, what is this fundamental age-related break that's resisting the conversion?
The multiomics work was very, very clear on this. The core barrier is cellular senescence.
Senescence is a concept our listeners are really interested in, so let's nail down what that actually means here. In the context of a heart scar, it's not just about being old, right?
No, not at all. It's much more specific. Senescence is a persistent stress response. These cells, they stop dividing, so they're not really adding to the scar by making more cells.
Okay.
But they don't die either. They just enter the state of metabolic rigidity. Their DNA gets kind of locked down, and they activate this devastating signaling pathway. It's called the senescence-associated secretory phenotype, or SASP.
That sounds like a chemical keep-out sign that the cells are putting up.
That is a perfect way to put it. It is a toxic cocktail of hundreds of secreted inflammatory factors. We're talking cytokines, chemokines, enzymes, and these senescent cells just continuously pump this stuff out. It makes the local tissue environment completely hostile to any attempt at regeneration.
The researchers obviously had to prove this link, that senescence is what causes the reprogramming to fail. How did they actually confirm that?
They used 2 really clean lines of evidence. First, they just looked at the aged cells under a microscope. About 30% of those aged cells stained positive for SABOG. It's a classic marker that shows that stiff senescent state.
And the young cells?
Only 2%.
So more senescence equals less regeneration. It's a direct correlation.
Exactly. And second, they took those superplastic young cells and they artificially forced them into senescence. And when they then tried to apply the MGT cocktail, the efficiency just tanked. It went from that baseline of 15% right down to the age level of 2%.
That's definitely the cause.
It's the cause. Senescence, whether it's natural or induced, is the primary enemy of reprogramming.
That makes perfect sense. So if senescence is the barrier, the logical first step is to try and clear it out. What happened when they used senolytic drugs, ones designed to, selectively kill those senescent cells?
This was a really crucial proof of concept. They treated the aged cells with a senolytic drug combo. And it worked. It eliminated some of the senescent cells, and it boosted the reprogramming efficiency from that dismal 2% baseline up to about 6%.
6%. That's a threefold improvement. That sounds pretty encouraging.
It is, but here's the critical point. 6% is still nowhere near the 15% they got in the young cells. So this suggested that senescence wasn't just there. It was actively suppressing the entire process. And just killing the senescent cells wasn't enough. They had to find the molecular switch that was driving the senescence and suppressing the heart genes at the same time.
Okay, so proof of concept is down. Now we get to the molecular manhunt. Finding that single switch, how did they even begin to narrow down the search?
They performed a huge loss-of-function screen. So they systematically silenced hundreds of potential regulatory factors just to see which one, when you remove it, lets the fibroblasts jump that age hurdle.
They were looking for a saboteur.
A cellular saboteur, exactly. A factor that actively represses the heart program.
And who's the main suspect that emerged from this?
The star of the show is an orphan nuclear receptor called Nr4a3. When they knocked down Nr4a3 in those really stubborn aged CFs, they saw the most dramatic increase in new heart cell generation. The efficiency jumped threefold, from about 1.8% in the control up to 5.8%. It was clearly the main molecular break.
What's so fascinating here is that Nr4a3 is acting as this master repressor with a dual mechanism. It's not just making the environment bad,
right?
It's directly blocking the repair job itself. Let's start with its role in aging.
Well, Nr4a3 is a chronic promoter of that age state. Its expression just naturally goes up as cells get older. And when they took those healthy young fibroblasts and forced them to overexpress Nr4a3, the results were definitive. The cells immediately started showing senescence markers, and their reprogramming rate just plummeted from 13% down to only 4%.
So it actively pushes the cell towards that non-regenerative state.
Shoves it, yeah.
Okay, so it's the accelerator for aging. Now, what about the immediate environment, that toxic SASP cocktail we talked about?
It directly drives it. Nr4a3 acts as a transcriptional activator for the SASP. It literally turns on the production line for those inflammatory genes. We're talking targets like CXCL14, IL-1A, FAP. It's actively commanding the cell to pollute its own neighborhood.
And they showed that this local pollution is enough to stop the conversion.
Absolutely. When they just took an antibody and blocked one of those targets, SCL14, the reprogramming efficiency went up right away. It confirmed that the inflammatory signaling driven by Nr4a3 is a huge part of why the MGT factors just can't do their job.
Let's go deeper then to the epigenetic control. Is Nr4a3 just creating a toxic neighborhood, or is it fundamentally locking down the cell's identity?
It's both. And this is where the discovery gets really powerful. Think of the cell's DNA like a huge library of blueprints. Nr4a3 isn't just screaming inflammatory signals in the reading room. It's physically locking the doors to the most important blueprints.
The cardiac blueprints.
Exactly. They showed that Nr4a3 maintains a repressive chromatin state. It physically suppresses key cardiac transcriptional programs. Specifically, it binds directly to the promoter of a gene called Myocardin, or Myocd, and just shuts it down.
And Myocd is, that's the master key for heart development, isn't it?
It is the ultimate master regulator. If you can't turn on Myocd, you can't switch the cell's identity, period. So when you deplete Nr4a3, you release that epigenetic brake, myocyte expression surges, and the whole chromatin landscape shifts from a scarred state toward a regenerative cardiac one.
That is just, it's remarkable.
Exactly.
A single target that clears out the inflammatory baggage and hits the accelerator for the heart genes.
So what does this elegant strategy mean when you move out of the dish and into a living scarred heart?
Right, the in vivo test. They used a mouse model of MI. They delivered the standard MGT reprogramming factors, but this time they also added a virus designed to specifically knock down Nr4a3. And they used lineage tracing to be sure any new heart cells they found definitely came from the old fibroblasts. So MGT plus the inhibitor. What did they find at the cellular level?
They saw a really significant increase in the absolute number of new induced cardiomyocytes in that injured area. And what's more, those new cells weren't just, there. They were structurally superior. They showed much better sarcomere organization, which means higher quality functional muscle.
But the real test for you, for the listeners, is the clinical outcome. Did it actually improve heart function?
It did, dramatically. They tracked heart function with echocardiography 4 weeks after the heart attack. In the control group, the MGT-only group, the left ventricular ejection fraction, or EF, how well the heart pumps blood, it improved a bit, but it was still low, around 40%.
Which is often the threshold for heart failure symptoms.
Precisely. But in the combination group, MGT plus the Nr4a3 knockdown, the EF improved to an astonishing 55%.
Wow.
That is a massive functional jump. It's pushing the heart back toward a near-healthy capacity, and the scar size was significantly reduced too.
That level of recovery is profound.
Yeah.
And I have to ask, was this conserved in human cells? Does it work for us?
The essential validation, yes. They confirmed it in human senescent fibroblasts. When they knocked down the human version of the gene Nr4a3, it dramatically enhanced human iCM reprogramming. It increased Myocd expression. And when they grew these cells in a dish, they started to show rhythmic calcium flux and even spontaneous, sustained beating.
That's incredible.
It confirms that this Nr4a3 senescence barrier is a conserved roadblock in human physiology, which makes this so relevant for future therapies.
So if we bring this all together for everyone listening, what is the single most important takeaway from this deep dive?
I think the central insight is simple. The adult heart's failure to regenerate is, fundamentally, an aging problem driven by persistent cellular senescence. And this nuclear receptor, Nr4a3, is the master switch in that process. It locks down regenerative potential by both driving that chronic inflammation and by physically repressing master cardiac genes like Myocd. So, targeting Nr4a3 gives you the dual action strategy you need to clear the roadblocks and heal the aged, damaged heart.
It's amazing to think that finding that one repressive switch could unlock the full potential of this whole field.
And here's a final, maybe provocative thought for you to reflect on. While this research clearly shows that chronic, persistent senescence is bad, some evidence hints that transient short-term senescence might actually be needed to kickstart repair.
Ah, interesting.
Yeah, we see it in neonatal regeneration. So future research needs to figure out how we can precisely time the inhibition of things like Nr4a3 to block the toxic persistent effects, while maybe harnessing any of the beneficial short-term signaling. Getting that balance right could define the next decade of regenerative medicine.
That certainly gives us something fascinating to think about, the idea of perfectly timing cellular aging. Thank you so much for guiding us through this.
My pleasure.
And thank you for joining us for this deep dive into the latest research. If you enjoy the conversation, please remember to like, subscribe, and share. Keep supporting the SANS Science experience. We'll be back soon with another stack of incredible sources to explore.